All right, ladies and gentlemen, so I'm going to walk you through your mitosis drawings review worksheet today. So you should have two colored pencils with you or two pens, something of two different colors. And what we're going to do is we're going to walk through the stages of mitosis together. And as we do so, we're going to go ahead and draw in some chromosomes, see what they look like, see how they move throughout the stages of mitosis. We'll also label some important structures that you should be aware of, as well as put some major notes of what kinds of activities are happening during each stage of the mitotic division. This is going to serve as a really great resource for things like worksheets and labs, so great reference and also a good study tool for you. So let's go ahead and get started. So we're going to start with the first stage of mitosis, and that's prophase. So we've already gone through interphase, which means we've already duplicated our information. We've already duplicated our organelles, although we're not going to show those organelles on this particular worksheet. Know that they have been duplicated. We've gone through those checkpoints at the end of G1, we've gone through the checkpoint at the end of G2, and our cell has been given permission to go through the actual stages of mitosis. So remember that mitosis is the division of the nucleus. So really we're going to trace the division of the genetic information, so your DNA, your chromosomes, that are in that nucleus and how they move through the stages of mitosis so that we can actually make copies of ourselves. So I'm just going to pick two colors here and draw in those initial two chromosomes that we're going to work with. So again, I'm just going to pick two colors. So how about we go red and blue? Okay, you guys can use whatever colors you want. Just make sure that you're following through as you go. Okay, so there is our two chromosomes, or our four paired chromatids. Now, we are also going to come in here and we're going to label some structures as well. So when we look at this, what are we actually looking at? Well, if I take a look, these guys here, okay, in this sort of starburst thing, these are our centrioles. Okay, and those are going to be responsible for where our spindle fibers come from, and then ultimately organizing those spindle fibers so that we can actually move this genetic information around. If I look at this whole structure here, all right, that whole thing is a chromosome. Okay, and each chromosome consists of a duplicated chromatid, all right, or sister chromatids, right? So a whole chromosome structure, right, when I look at this whole thing, each arm or each side, each bend is a single chromatid. Together we talk about them as a chromosome at this point during cell division. All right. They are then held together, so sort of kind of by this imaginary little piece here, all right, which we're going to call your centromere. And you can kind of think of it almost like Velcro. It's protein structure that is holding those two chromatids together until we eventually allow the cells to break that apart and actually separate that genetic information, that duplicated genetic information, into the cell. And right now we're at the beginning of prophase. So I'm still seeing my nuclear membrane in place. Okay, so we haven't started breaking down that structure yet, although as we start going through the process of prophase, that nuclear membrane will start breaking down. We can then actually start to see these chromosomes, and that's going to allow us to start moving them around the cell. So what kind of major processes are happening here? We're going to want to write these down next to this part on your worksheet. So this is the longest phase of mitosis. There's a lot going on here in terms of getting those chromosomes together. We're ultimately going to start seeing the chromatin. Remember, that's the loose DNA that we need access to when a cell is in normal function during interphase. Well, we've got to get all of that coiled down and condensed into this chromosome structure, which is what's going to allow us to see it. So during interphase, that chromosome structure is all loosely bound, kind of think of it like a big ball of yarn, and then we're going to coil it all up into the actual skein and actually make it into a chromosome. And when we do that, it's now something that can actually be visible. Another way to think about it is stretching out the slinky, right? So we can play with a slinky, we can use it, we can play with it, it's all stretched out, it's a little harder to see maybe from a distance. And then when it's time to just move it so nothing happens to it, it doesn't get damaged, we'll compress that slinky back down into its smallest form, aka the chromosomes. Well, now it might be easier to see from a distance, plus it's much more organized and much easier to move. Those centrioles, all right, right now they're kind of seeing them at the beginning of prophase. They'll start moving apart from each other. There's two of them there. There's two different dots. And they'll start moving to opposite ends, or what we call the poles of the cell. And from those centrioles, we'll start having spindle fibers. We're not seeing those guys just yet. We'll see them in the next picture. But those spindle fibers will start stretching from those centrioles to the centromeres on the chromosome so that we can attach everything, make everything nice and aligned and easy to move, hopefully without breaking or losing anything. And by the time we get to the end of prophase, that nuclear membrane will disappear, and the nucleolus, which we're not seeing here in the picture, but that would be in the center of that nucleus, is also breaking down and disappearing. So all of that should be written down on your worksheet. If you need to hit pause here so that you can make sure you do so, make sure you hit that pause button, get all this stuff onto your worksheet. Once it's all there, you can go ahead and hit play again and move on to the next phase. Okay, so once prophase is completed, we're going to move on to metaphase, which is the next phase of our cell division. So we're going to go ahead and draw in those chromosomes again. Okay, they're still together at this point. They're still paired in those sister chromatids. So I'm going to draw this guy up here. All right, and we were using blue, so I will draw him down here. Okay, and we're going to go ahead and label some structures on this one again as well, so we know what we're looking at. So when I do that, all right, well now we can see that those centrioles, right, they were paired in the last one, have now moved to opposite sides of the poles, or opposite ends of the cell. So I've got one here and I've got one over here. All right, I still have these guys connected. Okay, so there's still in metaphase a centromere that is holding these guys together. Okay, and each one of these arms is still a chromatid or a sister chromatid. And I'll draw that centromere down here even though we won't label it. And I still have my whole chromosome structure here. Okay. But now we can actually see those spindle fibers, and they're stretching from those centrioles to those centromeres to be able to help us in the next stage to separate out those chromatids and hopefully do that evenly and effectively. Okay. So when we talk about what's going on here in metaphase, this is the shortest phase. Really, once we get to the end of prophase and all those spindle fibers are attached to all of our chromosomes, it's going to rapidly cause all of those chromosomes to just snap in place in the middle of the cell. And as soon as that happens and everything's connected and everything's lined up in the middle, we're good to go to the next phase. So those spindle fibers here at metaphase are really just finishing their attachment point from the centriole right down here, right down here to the actual centromere itself. All right, and that's really all that's happening here. Once we do that, those chromosomes then line up in the middle of the cell, and that middle sort of imaginary line is what we call the metaphase plate. All right, and so let's go ahead and draw that in because that's not going to hurt to have that drawn in there as well. So it's just this imaginary line here in the middle of your cell. Kind of think of it almost like the equator on the Earth. All right, and that is known as your metaphase plate. Okay. And it's just called that because that's where everything lines up when we get to this particular stage. Also, don't forget, this is another checkpoint. So we had a checkpoint at the end of G1 just to make sure that all the division from the previous cell division went okay, that the cells were actually getting big enough to need to divide again. We had a checkpoint at the end of G2 to make sure that genetic information got duplicated correctly, that our organelles got duplicated, and then here is the third one. So this happens here at this point because we want to make sure that all of those spindle fibers are properly attached to all of those centromeres, because the next stage here is to actually start pulling those sister chromatids apart and separating out that duplicated genetic information. We want to make sure that that split happens evenly and accurately so that the cells that we create at the end of mitosis actually have all of the genetic information that they need to be a functional cell. Okay, so we're now on to anaphase, and this is really going to be where we start splitting out that information. So I'm going to start moving those sister chromatids to opposite sides of the poles. So they're now split. They're no longer connected to each other. And so we're going to see that split. And it's going to look something like this. And if we go back and label some of those important structures again, let's see where they have ended up. So I still have my centrioles here. Okay, and again, there's one on either side of the cell. I still have those spindle fibers, but notice that they're getting shorter. All right, and they're getting shorter as we continue to move those chromatids to opposite sides. Now here's where things get a little funky. Once we get to this point, you could still call this a chromatid. And some websites, some textbooks, some people you talk to will still call them sister chromatids. But you may also come across it as an individual chromosome at this point, once it's detached. So unfortunately, it's one of those weird things where there isn't necessarily consistency, depending on who you talk to, what text you come across. So just know you could kind of use these terms interchangeably at this point. Once they are no longer connected, that centromere, or excuse me, that centromere is no longer there. It's gone. That Velcro has basically come apart. And so they may refer to them as individual chromatids still, or you may come across it as a chromosome. So just kind of be careful to look at the context in which you're looking once you get to this point. So if we put some notes off to the side as to what's happening, here your sister chromatids are separating. So that centromere is essentially broken down. We've kind of ripped the Velcro off, if you will. And those chromatids or chromosomes, depending on what term you come across, they start moving to opposite ends of the poles, and they're going to move along those spindle fibers just to ensure that this separation of that information happens evenly. And then our final step here is telophase. So here we're showing the cells at the end of telophase. So the split has been completed. All right, we've actually finished our cytokinesis. All right, and we're going to actually go ahead and show what these cells look like once cell division is complete. And what you're going to see is that you've got one type of chromosome, all right, in this case the colors that we're using. There should be one of each in each daughter cell that we create, assuming that this has happened correctly. And so you should have one of each. Okay. In addition to that, some other structures will come back at the end of this. So these chromosomes will be within a nuclear structure again. That nuclear structure is going to come back. Okay. So if we look here, this is a nuclear membrane. not only will the nuclear membrane come back, but the nucleolus will come back as well. All right, and we now talk about these as individual chromosomes, regardless of kind of who you talk to at this point. By the time telophase and cytokinesis are finished, they will call those individual chromosomes at this point because they are now in separated cells. So telophase in and of itself is the last step of mitosis. What you're seeing here is both telophase and cytokinesis. So we're showing you at the end. If I were to show you true telophase, those cells would still be connected. You just have two nuclei or two nuclear structures in a single cell until the cytokinesis finishes splitting out the cytoplasm. So your centrioles and your spindle fibers at this point have disappeared because they've kind of done their job moving that genetic information, moving those chromosomes to opposite ends. The nuclear structure comes back. So again, we drew in the nuclear membrane. The nucleolus would also come back, which would be in the center of that nucleus. Those chromosomes will also start relaxing back into chromatin. We're moving into another round of interphase for these two cells. And part of interphase, remember, is just your cells doing their normal day-to-day function. And so we need access to that genetic information to be able to tell the cells what to do. Well, in that chromosome structure, right, think of it like a condensed slinky. You really can't play with it. You can't do anything with it. And so we need to relax it back into chromatin, or that stretched out slinky phase in that analogy. And so your chromosomes will do the same. They'll relax back into that chromatin so that we can actually access the information. This stage, like I mentioned, will also overlap with cytokinesis, and what you're seeing here in the actual picture itself is at the end of cytokinesis, which is where we split the cell. The actual cytoplasm separates, and we form these two new daughter cells. And those two daughter cells, it's really important to remember, are identical. They're identical to each other, and if you look at it, right, you've got both a red and a blue in each cell. They are also identical to the original parent cell prior to that parent cell going through the duplication of its genetic information. So all of these cells are genetically identical to each other, and they're genetically identical back to the original parent cell. So when we go to, say, make new skin cells because you got a paper cut and we need to fill in and replace those damaged cells, we want them to look and function exactly the same as the original cells that were there. And that would be a purpose to mitosis. You're getting bigger, so we need to make more muscle cells. Same deal. I want those muscle cells to act just like the original parent muscle cell, so all I want to do is make a copy of it, and mitosis is the way to do that.